Right. So starting off with the topic of photosynthesis. So we know photosynthesis, of course, is the process that is done by plants where they trap light energy and they make their own food. So it basically they you have your carbon dioxide. Carbon dioxide reacts with water to give you glucose and oxygen. So in this topic, we will study in detail exactly how that is happening. Now, when we go ahead and we talk about it, so uh, uh, we energy is constantly flowing through our ecosystem, and organisms can be classified according to how they're getting their energy, right? Autotrophic organisms or autotrophs, if you look over here, they are organisms that can make their own organic compounds from carbon dioxide, mostly by the process of photosynthesis. They capture energy from the sun, they turn it into chemical energy, and uh, um, they turn it into chemical energy, and that chemical energy will lead to the formation of glucose, and glucose can then be turned into starch, right? When we go ahead and we talk about it, there are certain organisms that are considered to be heterotrophic. Heterotrophic organisms generally eat plants or other animals which have eaten plants. So basically, if you remember your IGCSE, that would be your primary consumers or your secondary consumers. So heterotrophs are your primary consumers and your secondary consumers, while autotrophs are often considered to be your producers. Now, when we talk about making chemical bonds, because that is what I just told you, that autotrophs will take light energy and they will turn it into chemical energy, and they will utilize the energy to make bonds. When we talk about making chemical bonds, you need to have an input of energy. Energy constantly has to be available in an easy form, in an accessible form, so it can be instantly available for use for multiple different reactions. Now, there is a simple molecule that does all of this for us, right? That molecule is our ATP, adenosine triphosphate. This is the structure of ATP. We have our ribose sugar, fine. The ribose sugar is connected to adenine. Adenine is our nucleic acid. The same nucleic acid that we used to have in DNA, that exact same nucleic acid is connected to our ribose sugar. And then it is connected to inorganic phosphate head, specifically 3. That is why it is known as adenosine triphosphate. That is the difference between ATP and adenine. Adenine being the nucleic acid base that we have in DNA. In DNA, when we have our adenine, that consists of ribose and one phosphate head. Or in DNA, it was deoxyribose and one phosphate head. In ATP, we have ribose, we have adenine nucleic acid, and we have 3 phosphate bases. Whenever energy is needed, the third phosphate head can be broken by a hydrolysis reaction. So what can we do? We can do a hydrolysis reaction where we can take our ATP and we can break it down to make ADP. ADP is adenine, adenosine diphosphate. Because I have broken one of the phosphate bonds. I have broken one of the phosphate bonds. I have removed it. And when I'm going to break the bond, energy is going to be released. So my molecule is going to break down. It's going to hydrolyze. It's going to turn from ATP into ADP. And in this process, energy is released. The amount of energy released is 34 kilojoules. 34 kilojoules of energy is released per mole of ATP that is being hydrolyzed. So this is exactly how energy is available for different processes. right? This is what is taking place. So this is our ATP in the presence of water and our enzyme. The enzyme is ATPase. So in the presence of water and our enzyme ATPase, ATP can be turned into ADP. The third phosphate head can be broken, and in the process, energy is actually released. This process is pretty much reversible. Okay, so what will happen that the ATP is going to break down into ADP and a phosphate head. Then the ATP can be remade. ATP can be synthesized again. You can take the ADP and you can add the phosphate group to it and turn it back. You can again do a condensation reaction and turn it back into ATP. So it's a reversible reaction. You can break it down to give you ADP and phosphate head that will release the energy. Once the energy is released, the ADP can combine back with phosphate head to give you ATP through a condensation reaction, right? Now, to make ATP, to do the condensation reaction, to join the ADP back with the phosphate head, we need energy. Where does this energy come from? This energy comes from the reduction and oxidation reactions that are taking place inside the cell. So inside the cell, we have reactions taking place, redox reactions. Those reactions will provide the energy so that ADP can combine with the phosphate group and give you ATP. So ATP, breakdown of ATP gives us energy. To combine ADP back with the phosphate group and again make ATP, for that we need energy. So that energy will come from the redox reactions, and that's what we're going to study. How do we make this ATP? How is this ATP made? Right? Where are we getting this energy to combine the ADP back with the phosphate group and give us ATP? Where are we getting that energy? That energy comes from something known as the electron transport chain. The electron transport chain is found both. It is found in our plant cells and it is found in human cells as well. I mean, animal cells as well, right? In the mitochondria and in the chloroplast. In both of them, you have your electron transport chain, but the process is different. In mitochondria, the process is different. In our plants, the process is different. But of course, we don't need to worry about the mitochondria one right now. That we will cover later on. So when we talk about the electron transport chain, so there are two main ways in which ATP can be formed, right? One is using energy released from catabolic reactions which are taking place. For example, a cellular respiration. So in cellular respiration, you're doing a catabolic reaction, breakdown reaction, and in the reaction, energy is being released. So you can use that energy to make ATP. That's one way. But the actual main way is by using the electron transport chain, where you do removal of hydrogen atoms from some intermediate compounds. So we have some intermediate compounds, and from those intermediate compounds, we generally remove 2 hydrogen atoms, right? These hydrogen atoms are removed from the compound. The hydrogen atoms are then collected by a hydrogen carrier. A hydrogen carrier will then collect these hydrogen atoms. Okay, you can say hydrogen carrier or hydrogen acceptor, both are correct. Okay, you can say hydrogen carrier or hydrogen acceptor, both of the statements are correct. So we have a hydrogen acceptor. The hydrogen acceptor will accept the hydrogen atoms. In the process, the acceptor will be reduced because you are taking hydrogen atoms. Gain of hydrogen atoms is reduction. We know that from chemistry. So the acceptor will take the hydrogen atoms and the hydrogen acceptor will be reduced. The electrons from the hydrogen atoms are then transferred along a series of carriers, and this series of carriers are known as electron transport chain. So 2 hydrogen atoms are produced or removed from the compound. The 2 hydrogen atoms will pass on to the hydrogen acceptor. The acceptor will be reduced. and the electrons from the hydrogen atom will then be carried across the electron transport chain. So as the electrons will move in the chain, the components of the chain, the different parts of the chain, we will study that. Different parts of the chain will be reduced, and they will be so when they will receive the electrons, when they will get the electrons, so when the electron is going to pass from one carrier to another, so when they will receive the electron, they will be reduced, and when they will transfer the electron to the next carrier, they will be oxidized. So what will happen, that the same carrier will be reduced when it is taking electron, and the same carrier, when it is passing the electron to the next one, it will be oxidized. So in this redox reaction, small amount of energy is released, and this energy is then used to make ATP. This is just a general overview. We are going to study in more detail how the process is taking place. But generally, we have compounds. From the compounds, we remove hydrogen atoms. The hydrogen atoms will pass on to the hydrogen acceptor. The hydrogen acceptor will be reduced, and the electrons from the hydrogen atoms will pass on to the electron transport chain. Now, in the electron transport chain, as the acceptor will take the electrons, it will be reduced. And as it will transfer the electron to the next portion, it will be oxidized. In these redox reactions that are taking place, energy is released, and that energy is used to make ATP. That is a basic overview of how the electron transport chain works. We are, of course, going to study in detail how it is happening, but that's a basic overview. Any confusion? Is this making sense?
Yes, miss.
Okay. Now, when we talk about our photosynthesis, this is the equation you know perfectly well. Carbon dioxide reacts with water in the presence of light energy to give us glucose and oxygen. Now, what you need to have a good enough idea of is the structure of chloroplast. This is something you might have covered a little bit in your unit 2 when you did structure of your plant cell. You might have studied this, but now you need to study this in a lot more detail. So first thing that we know about chloroplast is basically surrounded by, it's a double membrane organelle, right? It will have an outer membrane as well as a inner membrane. There is a space between the outer membrane and the inner membrane. The space between the two membrane is known as the chloroplast envelope. Now, when we talk about it, inside the chloroplast, we have a system of membranes. The membranes are arranged in layers. The layers are, so if you look over here, this. We have membranes. The membranes are arranged in layers. It looks like you have a stack of coin, right? So it is arranged in layers, and the layers are known as grana or granum. Granum is plural. A singular. Grana is plural. A single granum is made up of layers of membrane. So that's what I told you. We have layers of membrane that are stocked on top of each other to make our grana. Inside one granum, we have these membranes. The membrane is known as thylakoids. So basically we have a membrane. The membrane is thylakoid. The thylakoid is layered on top of each other to give us a stack of points. And that stack of coin is known as our granum. Inside the thylakoid membrane, that is where the green pigment chlorophyll is going to be found. The grana are joined together to each other by lamella. So lamella are going to be over here. If you look, this is lamella. Lamella are going to be joining the grana with each other. What are lamella? Lamella is the extension of your thylakoid membrane. So the extension of the thylakoid membrane is known as the lamella, and lamella is going to be joining the grana with each other. Now, around this entire structure inside the chloroplast, we have a matrix. The matrix is known as stroma. Okay, the matrix is known as stroma. Now, when we talk about the thylakoid, inside the thylakoid, I told you we have the chlorophyll. So, inside the thylakoid membrane, that is where light energy is going to be captured. Then we have the stroma. Inside the stroma, we have enzymes that are going to complete the process of photosynthesis and actually produce glucose. So glucose can then be used for, respiration, storage, whatever you want to go ahead and do with it. Now, when we talk about chlorophyll, right? Chlorophyll is, it is composed of different pigments. So you can have chlorophyll A. Chlorophyll A is blue-green in color. You can have chlorophyll B, which is yellow-green. And you can have carotenoids. Which is orange, carotene, and yellow xanthophyll. Just give me 1 second, Manur, okay?
Okay, Miss, take your time.
Right. So when we talk about our pigments, chlorophyll A is going to be found in all photosynthesizing plants, right? Chlorophyll A is found in all photosynthesizing plants, and it is the highest in quantity from all our pigments. From all the pigments that we have, this is the one that you're going to find in highest quantity. And if you take a look at our absorption spectrum, right? If you take a look at absorption spectrum, you will go ahead and you will observe this. 1 second, let me clear this up. You're going to go ahead and you're going to observe exactly this. So the absorption spectrum describes the different amounts of light of different wavelengths that the photosynthetic pigment absorbs. So if you look at the graph, you will clearly see what? That chlorophyll A is the one that is found in greatest quantity. Then along with that, we have chlorophyll B, we have carotenoids, and we also have phaeophytin. Phaeophytin is a gray pigment, phaeophytin. Phaeophytin. So these are the different pigments that you need to go ahead and you need to know about. And this is basically when you can do paper chromatography, you will observe these results, right? So if you do paper chromatography for the pigments, these are the results that you're going to observe. So you can see all of these pigments. The xanthophyllene and the carotene is collectively known as what? Carotenoids. You have phaeophytin, which is gray, chlorophyll A and chlorophyll B. Now, whenever you do paper chromatography to actually separate the components, what is going to happen? So plants look green in color, right? But if we extract the pigments from the plants, we will grind the leaves, and then when we're grinding the leaves, we will first add propanone to it. Okay, we will add propanone and we will grind up the leaves. Then we will filtrate it, and after doing the filtration, we will obtain the filtrate. After doing the filtrate, we will do the process of paper chromatography. So in paper chromatography, we will take our chromatography paper or filter paper and we will mark the baseline with a pencil. On top of the baseline, we will use a dropper, or you will add a drop of our pigment, of our chlorophyll pigment. How did we get the chlorophyll pigment? We ground up the leaves with propanone, we filtered it, and we used the filtrate. So we will add the filtrate into the baseline. After that, you will place the paper chromatograph into a solvent, any solvent, and you will leave it. Now, what will happen that when you talk about your chromatography, so different pigments have got different adherence to the paper and different solubility in the solvent. Those pigments that are more soluble in the solvent, they will travel further along. Those that are less soluble in the solvent and they have greater affinity for paper, they will travel a lesser distance. So what are you going to do? You are going to let the process happen. The different pigments inside the chlorophyll, they will all end up separating. They will all end up traveling different distances because they all have different solubility in the solvent. Now, what you can do is that you can calculate the RF value, retention factor value. How do we calculate the retention factor? Distance traveled by the solute or our pigment divided by distance traveled by the solvent. So the solvent would have traveled all the way through the paper. So using a pencil, you can mark how much distance was traveled by the solvent at the end of the experiment, right? That will be your denominator. In the numerator, you're going to go ahead and you're going to write down the distance traveled by the solute. That is going to be your pigment. You can divide the two of them to calculate what your retention factor is going to be. That will help you identify what the pigment can possibly be. Any confusion so far? Anything you want to ask?
No, Miss, I understood.
Okay. These are the terminologies. Okay, actually, before this, we need to talk about photosystems, right? There are two photosystems that we need to talk about. We have photosystem 1 and photosystem 2. Each photosystem has a different combination of chlorophyll pigments. Photosystem 1 is going to have pigments that absorb light of wavelength 700 nanometers. So those chlorophyll pigments that are absorbing light of 700 nanometers, those are going to be found in the photosystem 1. In the photosystem 2, you will find chlorophyll pigments which absorb light of wavelength 680 nanometers. Now, photosystem 1 is going to be involved in both cyclic and non-cyclic photophosphorylation. What is photophosphorylation? Photophosphorylation is when you do phosphorylation in the presence of sunlight. I will discuss the process with you. So there are two types of photophosphorylation that take place. We have cyclic photophosphorylation and non-cyclic photophosphorylation. So both of the systems or both of the processes will take place in photosystem 1. In photosystem 2, only non-cyclic photophosphorylation is going to be taking place. So this is basically what is happening. So when we talk about our photosynthesis, right? Photosynthesis takes place in two stages. We have light dependent stage and light independent stage. Light dependent stage is the one for which we actually need the light energy. The light independent stage does not require the presence of light energy. So when we talk about it, in the light dependent stage, and we have the light independent stages. So photosynthesis occurs in these two stages. So let's go ahead and take a look. When we talk about the light dependent when we talk about the light dependent stage of photosynthesis, so in the light dependent stage of photosynthesis, it occurs in the thylakoid membranes where you have the chlorophyll. That is where the light dependent stage is actually going to be occurring. There are two main functions or two main stages. The first stage is the breakdown of water molecules in a photochemical reaction. What do I mean by photochemical reaction? That I'm going to be using light energy to actually perform a chemical reaction. I'm using light energy to break down my water molecule to give me hydrogen ion, right? I will end up getting my hydrogen ions, as you can see in the diagram. Along with that, the second function is that it will be producing ATP, which supplies the energy that you need to build the carbohydrates. So to actually build carbohydrates and to actually make the glucose, you need energy. That energy is going to come from ATP. Where is that ATP coming from? That ATP is coming from the light dependent reaction. So light dependent reaction is going to be giving you what? It's going to be giving you hydrogen ions. Hydrogen ions is basically going to reduce your carbon dioxide. The hydrogen ions produced will reduce your carbon dioxide, and they will then help you make the carbohydrates that you need. So ATP and hydrogen ions are made in the light dependent stage. In the light independent stage, the carbon dioxide is reduced by the hydrogen ions, and the energy from the ATP is used to make your carbohydrate, that is going to be your glucose. So when we talk about the entire process, the cycle that is actually used is known as Kelvin cycle. The Kelvin cycle is the one that is going to be actually providing you your, you can say, glucose. It's going to allow the final production of the glucose. This occurs in the stroma. The stroma of the chloroplast, that is where the Kelvin cycle actually occurs. So, continuing with our light dependent stage. Continuing with our light dependent stage. So when we talk about light, light is actually made up of particles known as photons, right? When we talk about photon, what is a photon? Photon is basically the unit of light that contains energy. Now, what happens that when this unit of energy will hit our chlorophyll molecule, so inside our chlorophyll molecule, we have electrons because everything is made up of atoms, right? And inside our atoms, we have electrons. So when it will hit the chlorophyll molecule, the energy is transferred to the electrons of the chlorophyll molecule. What happens? This is a little bit of physics. The chlorophyll electrons in the chlorophyll molecule gain energy. And as they gain energy, they go from a lower energy level to a higher energy level inside the atom. as they will go to a higher energy level inside the atom. So what is going to end up happening, that if the electron gains enough energy, it will end up leaving the chlorophyll molecule completely, because it will become delocalized. This delocalized electron or excited electron will then be collected by an electron acceptor. An electron acceptor will take this electron, and this electron will then be used for cyclic photophosphorylation and non-cyclic photophosphorylation. Just 1 second. So when we talk about the synthesis of ATP, so it will be passed on to the electron acceptor. And once it is in the electron acceptor, it is going to pass on to two stages: cyclic photophosphorylation and non-cyclic photophosphorylation. Now, both of the processes will take place at the same time. In a cyclic photophosphorylation, what is going to happen? One second. Right. So this is what is happening in cyclic photophosphorylation. So in cyclic photophosphorylation, we have our chlorophyll. Light energy is going to fall onto our light energy is going to fall onto our light energy is going to fall onto the chlorophyll molecule. Inside the chlorophyll molecule, we have what? We have electrons. The light energy will be transferred to the electrons, and the electrons will become excited. As the electrons are going to end up becoming excited, they will jump from a lower energy level to a higher energy level. And as they will go to a higher energy level, they will become delocalized. They will become excited. From there, they will pass on to this a series. It will literally be a series. We will have a series of what? We will have a series of electron carriers. And that series of electron carriers will pass on the electrons from one to another. As they will pass on the electrons, what will happen? When they will receive the electrons, they will be reduced. When they will transfer the electrons, they will be oxidized. This redox reaction will produce energy that will allow your ADP to combine with the phosphate group and give you ATP. And then the electron carriers will take those electrons and they will return it back to the chlorophyll molecule. So this is what is happening in your cyclic photophosphorylation. Okay, this is phosphorylation. Why? Because you're adding a phosphate group. Phosphorylation is when you're adding a phosphate group. Since you're adding a phosphate group, this is phosphorylation. Then when you're going to go ahead and talk about it, we are using light energy to do it. So photophosphorylation. And why is it cyclic? Because it is returning the electrons back to the chlorophyll molecule. This is only done in photosystem 1. It is only done in photosystem 1. And this is going to what? This is going to lead to the production of ATP. And once the molecule electron is back in the chlorophyll, it will again start the process. Then we have non-cyclic photophosphorylation. In non-cyclic photophosphorylation, what is done? That water molecules are broken down, providing hydrogen ions that you need to reduce NADP. Right? NADP is one of the compounds that is used. So to reduce NADP, we need to go ahead and we need to get hydrogen ions. Those hydrogen ions are going to come from the breakdown of water. So when we, and in this process, ATP is again going to be produced. Now, this non-cyclic phosphorylation is done both in photosystem 1 and photosystem 2. This is non-cyclic because the electron does not return back to the original system. Now we will start over here from photosystem 2. So what is going to happen? In the light, photons constantly hit the chlorophyll molecules in both photosystem 2 and photosystem 1. The electrons will get excited to a higher level. As you can see over here, they're getting excited to a higher level. They are the electrons are lost from the chlorophyll molecule and they are passed on to the electron acceptor and transferred along the electron transport chain to photosystem 1. So from photosystem 2, they will electron will get excited. It will pass down the electron transport chain to photosystem 1. Now, what is going to happen? That as the electron is going to pass from the electron transport chain, it will provide the energy needed for the formation of ATP. So ATP is going to be produced as the electron moves down the electron transport chain. Now, the photosystem 1 will receive an electron to replace one that was lost to light independent reactions. The chlorophyll molecule in your photosystem 2 is missing an electron, so it is unstable, right? It is missing this photosystem 2, it has lost an electron. It has lost an electron because the electron has gone to photosystem 1, and this is non-cyclic. So right now, photosystem 2 is unstable because it has lost an electron. So what is going to end up happening? That the electron has to come back. We somehow have to bring the electron back to photosystem 2. So we are going to do that by photolysis. We will break down a water molecule to give us hydrogen ions and hydroxide ions. So because of that, what is going to happen? That the electrons produced will replace the electron in photosystem 2. The electron, so the entire process, chlorophyll will absorb light energy, electrons will get excited. As the electrons will get excited, they will travel down the electron transport chain and they will produce ATP. They will produce ATP and the electron will pass on to photosystem 1. The electron that has been passed on to photosystem 1, what is going to end up happening? That those electrons will transfer to NADPH, NADP, and the NADP is going to become reduced and it will turn into NADPH. So in this entire process, the electron has moved from the photosystem 1 to photosystem 2. So now photosystem 2 does not have, is having one less electron. To replace those electrons, we will break down a molecule of water, which is known as photolysis, and the electrons released in the process will replace the electrons that were lost by photosystem 2. Now, while this entire process is happening, the light energy will also fall on photosystem 1. So the electrons in photosystem 1 will also become excited and they will be collected by an electron acceptor. They will be transferred down the electron transport chain and they will be collected by NADP as well. The NADP is going to then get reduced and it will turn into NADPH. So the reduced NADP or the NADPH that is being produced and the ATP provide the source of, they provide the compounds that you need to actually make glucose. So in this entire process, what is going to happen at the end of both cyclic photophosphorylation and non-cyclic photophosphorylation, you are going to end up with ATP and reduced NADPH. Both of these are then going to be used for what? Both of these are then going to pass on to the stroma of, this is all happening in the thylakoid membrane. It will pass to the stroma of the chloroplast where light independent reaction will take place, that is known as Kelvin cycle. Anything confusion so far? Is this all clear?
Yes, Miss, it's all clear.
Okay. Now let's talk about the light independent stage. Okay, so in the light independent stage, basically what is happening that the NADPH and the ATP will pass on to the stroma and they will enter into your Kelvin cycle. In the first step of Kelvin cycle, carbon dioxide from the air will combine with ribulose biphosphate. This is ribulose biphosphate. It is a compound that has 5 carbons in it. So ribulose biphosphate has 5 carbons in it. This will combine with your carbon dioxide. This process is known as carbon fixation. Fine. The carbon dioxide is said to be fixed. So this is known as carbon fixation. Now, what is going to end up happening? This step is done with the help of an enzyme. The enzyme is known as ribulose biphosphate carboxylase, or you can use a short form, Rubisco. Right? So Rubisco is the needed for this carbon fixation to take place. And this is the rate limiting enzyme in the process. Okay. So if Rubisco is saturated, the rate of reaction will be limited. Now, what is going to end up happening that these two will combine with each other to give you a 6 carbon compound. And then you don't have to worry about the 6 carbon compound. The 6 carbon compound will instantly break down to give you glycerate 2-phosphate. Glycerate 2-phosphate is a 3 carbon compound. So you will end up with 2 molecules of glycerate 3-phosphate. So your ribulose biphosphate will combine with carbon dioxide to give you a 6 carbon compound. The 6 carbon compound quickly breaks down to give you a 3 carbon compound, that is glycerate 3-phosphate, and 2 molecules of glycerate 3-phosphate are going to be produced. After that, now remember that reduced NADPH that had hydrogen ions on it, right? So what will happen that the hydrogen ions from the NADPH as well as the ATP, the energy from the ATP is going to be used for reduction. The glycerate 3-phosphate will be reduced to give you glyceraldehyde 3-phosphate. Again, 2 molecules of glyceraldehyde 3-phosphate will be produced. Now, when we go ahead and we talk about it, the glyceraldehyde 3-phosphate is then converted to give you your glucose. Right? It is then going to go ahead and it is going to give you your glucose. And then some of it will be turned back to give you your ribulose biphosphate, and the process will start all over again. So this is how the process is actually taking place. This is how your Kelvin cycle is taking place. You're starting with ribulose biphosphate, which is the 5-carbon compound. The ribulose biphosphate is combining with carbon dioxide to give you, in the presence of rubisco, to give you a 6-carbon compound. The 6-carbon compound breaks down to give you 2 molecules of glycerate 3-phosphate. Glycerate 3-phosphate in the presence of NADPH as well as ATP will be reduced to give you glyceraldehyde 3-phosphate, again 2 molecules. After that, the glyceraldehyde 3-phosphate will then go ahead and it will be turned into glucose. So when we go ahead and we talk about it, this is light independent stage because we don't need light for this particular process. So this is what is actually taking place. So in this particular topic, you need to know your, you need to go ahead and you need to have an idea of what? You need to have an idea of your cyclic photophosphorylation, non-cyclic photophosphorylation, and the Kelvin cycle. You also need to know about your pigments. Now let's quickly go over limiting factors. Limiting factors are nothing new. You have covered limiting factors in your IGCSE as well. So this is not going to be anything much different, right? When we talk about limiting factors, one of the limiting factors can be light intensity. Light intensity affects the amount of chlorophyll which is excited and therefore the amount of ATP and reduced NADP that will be produced. If there's low light intensity, so less NADP and less ATP will be produced, and because of that, less glucose will be produced. Light is therefore a limiting factor for the process. And as we increase the light intensity, what will happen? The rate of photosynthesis will increase until you reach a plateau. When you reach a plateau, at that point, light intensity is no longer a limiting factor. When you reach a plateau, light intensity is no longer a limiting factor because increasing light intensity is not increasing the rate of photosynthesis anymore. Another thing it depends upon is carbon dioxide, right? Because if you don't have enough carbon dioxide for carbon fixation in the Kelvin cycle, the less glucose will be made, and as a result, carbon dioxide concentration will be a limiting factor. Then temperature. Temperature is very important because we have enzymes like rubiscose. Higher the temperature is going to be, higher the temperature that you are basically going to have, the more easily your enzymes are going to get denatured, because enzymes have like a tertiary structure. They are globular proteins. They have hydrogen bonds, ionic bonds, hydropic interactions. They have disulfide bridges. So what will happen that when you increase the heat energy, when you increase the temperature, the tertiary bonds are affected. Since the bonds are affected, the shape of the active site will change. The enzyme will denature. The substrate, ribulose biphosphate, will no longer be able to fit into the active site. And as a result, you will your enzyme substrate complex will not form and the product will not be made. If the temperature is too low, what will you observe? There's simply not going to be enough heat energy. So enzymes will not have enough kinetic energy. So there will be less collisions and there will be less successful collisions. So you need to have an optimum temperature, right? You need to have an optimum temperature, and at that optimum temperature, you are going to basically end up observing you're basically going to end up observing the maximum rate of photosynthesis. So light intensity, carbon dioxide, temperature, all of these can act as limiting factors to our rate of photosynthesis, and we will observe this on the graph as well. And at whatever point we reach a plateau, that factor is no longer the limiting factor. So if you look over here, like this is at a lower carbon dioxide concentration, this is at a higher carbon dioxide concentration. So at a higher carbon dioxide concentration, the rate of photosynthesis is much higher. And that is basically what we have to related to the topic of photosynthesis. Any confusion, anything you want to ask so far? Is this all clear?
Yes, Miss, it's all clear.
Okay. Okay, then Mahanur, we'll go ahead and we will stop it over here for today. Okay, tell me one thing. Do you want to do past paper? 1 second.